such a kind introduction, and hello to so many people I know there. I wish I were there in person. It's really was great to just chat for a moment before this talk. So I am going to cover today pretty much what Vivek just introduced. Let me go right to the next slide here and see if I can get that advanced. There we go. And sort of talk about, maybe this is almost the end of the talk really, which is where we're somewhat stymied right now on trying to figure out how to treat the tauopathies. There is tau antibodies, which have sort of risen to the level, have risen to the level already of a clinical trial, which failed, not surprisingly. We can talk more about this. I don't think it's a particularly good idea. There are antisense oligonucleotides, which may be interesting if we want to lower tau levels in general or go after mutant tau. It's an interesting possibility, although there was some disappointment there on a recent failure of the Huntington's disease trial with an oligo approach. There is this PROTAC approach, which is interesting. We have a lot more to learn. I'll say something about disaggregators, but most of what I'm going to talk about is trying to target the problem of tau spread, where you see that red arrow. And we'll go into that in some detail, beginning with some published data recently about discovery of the LRP1 receptor, and then to some new stuff. I doubt that I'll have time to talk about the last one. It is published about maybe a year and a half ago. You can look it up. Another approach to inhibiting tau degradation, to enhancing tau degradation by inhibiting farnesyltransferase inhibitors, as sort of unique approach, which we're also continuing to work on. So I think here's a slide that it's nice to start in a way that's familiar to people, and this is familiar to people. We know that tau does spread through the brain. This is the Brock staging, where it often begins in places like the entorhinal cortex and then spreads through the cerebral cortex. And this is the pattern we see in AD. But really, all the tauopathies spread. There is this prion-like process, with, I'm going to put the emphasis on the word like, there we don't have the worries that we do with prion disease itself in terms of that type of transmissibility. But it is prion-like in that the misfolded protein, tau as a misfolded protein, can template misfolding in neighboring cells. I'll speak a lot more about that. But I want to make a point here that it's not just spread in the Alzheimer pattern, but if you look over here at tau deposition in chronic traumatic encephalopathy, there's also a pattern of spread in which tau first forms at the depths of the sulci and then begins to spread from that point onward. And other tauopathies have their unique patterns of tau deposition and tau spread. So here's what we'll be talking about quite a bit, which is that there is misfolded tau arising in some cell in the brain. It could be arising because of CTE, because of trauma. It could be arising if you have a type of frontotemporal dementia with a tau mutation. There are many. It could be arising for, maybe it is somehow triggered with amyloid in Alzheimer's. We don't know why tau actually forms in Alzheimer's yet, but it's misfolded in cells, and then it will go to a recipient cell, as I mentioned, where the protein itself will template the misfolding of the normal protein in the recipient cell. So what is the conformation of tau as it passes between cells? And that's something we're very interested in. So to begin with stuff that's a bit more familiar with some published work here, we added tau both as monomer, oligomer, and even fibrils to cells in culture. The tau was fluorescinated, so we can look at its uptake and then use that as a measure. You can see those colorful lines up in the upper right show the amount of fluorescence of tau uptake in these cultured cells. We've used different types of cells. In this case, it was H4 cells, and tau is clearly taken up in these cells. This is work that was done by Jenny Rauch. And when it's, and so that gives us a nice system for looking at inhibitors of tau uptake. And the first thing we discovered actually a little while ago now was that tau, we could inhibit tau uptake by inhibiting the some modifications and chain extenders on HSPGs, heparan sulfate proteoglycans. As you know, they're sticking out from the cells all over the place. They sort of create this network in which ligands can glom onto receptors nearby, and by modulating the HSPGs, we can reduce tau uptake here. The arrow is pointing to a gene that was inhibited that reduced tau uptake and that extends the HSPG. But the discovery in this paper actually was that a specific modification on the HSPG, right there, this 6-OST, that modification, putting that sulfate in that position, really had a very nice effect on lowering tau uptake in recipient cells. But it wasn't completely satisfying, because we wanted to get a stronger inhibitor, and this is where we really hit the jackpot, because and then in trying some candidate genes, we ended up trying LRP1, and this is a member of a large family, which you see over on the right. There's a lot of relatives of LRP1 here that involves lipids, and it's a very well-known protein that has many, many ligands. It's kind of a trash can receptor for things that are floating around in extracellular space. And it looks like one of those things is tau, because if you go over here, you can see the inhibition. I think you see my arrow, the inhibiting LRP1 with the guide RNA. This is very different than the HSPG story. We just really knocked down the tau uptake to 0. It's a very impressive effect. And also equally interesting is that all these close relatives of tau, there's no effect on them whatsoever if we knock them down. So we have guide RNAs to all of them, and it was LRP1 that quite specifically, when that was inhibited, would knock down tau uptake. And it's a very big protein. It's very unwieldy. It's a little bit difficult to work with. You can see here in this panel here where it says LRP1 that it has all these different domains. I'll talk a little bit about that. And this was really stellar work that Jenny Rauch recently published. And just to give her a little plug, she's gone on and now gotten her own lab going at UMass in Amherst. But she continued this project, and I'll just maybe say one or two more quick words about this story, in which she went on to not just show this in culture, but to also show this in animals, in which she did this used this very nice construct that we got from Brad Hyman to Mass General, in which you have tau is fused to a GFP, and it's fused through this connector P2A. And what happens is, as soon as you put this into a cell, this P2A gets cleaved, and the GFP and the tau are now separate. So when we put this tau into the brain with a mutation at position 301, injected it into the brain, we can now see that if you look over here in the control experiment, the GFP up here stays in the same place, but the tau then spreads down through the brain. Whereas if you knock down LRP1 in the brain, both the tau and the GFP remain at the site of injection. So this also worked in animals. Now, the next question you may ask is, when it's spreading, where is it spreading to? Well, I mean, we can see where it's spreading to. It's spreading beyond the hippocampus and other regions of the brain, but what type of cells is it spreading to? And of course, it's probably spreading to neurons. That's, what we would suspect, but we also have the impression that microglia have a role here, because in the region where there's tau spread, we can get some labeling, not just of MC1, which is an antibody to tau in the recipient cells, but also in the area are these green IBA1 cells, or microglia. So microglia are becoming an increasingly interesting part of the story, and I'll be actually talking a little bit more about that coming up later. So just as I mentioned, one or two more quick things about this on the published work. The uptake through the LRP1 receptor seems to be mediated through the many lysines that are present here. Lee Gann did a proteomic study of tau and found all these lysines in tau, and lysines get acetylated. Many of them are acetylated. If we block the acetylation of the lysines, we can also reduce the binding of tau to the receptor. So blocking acetylation affects the tau uptake, because that's how the binding to LRP1 is mediated. Phosphorylation does not seem to affect uptake. The next question was, where in this monstrous LRP1 protein is the binding taking place? And just to jump quickly to the answer, it seems that if we make these what are we call mini LRPs, clone the individual domains, it looks like a lot of the action is right here in mini domain 4, which I'll just show you here. Here's the entire molecule, the entire protein, and you can see here that the part of tau, which actually ends up, according to many of the cryo structures we've seen recently, that's the part that actually is folded into the neurofibrillary tangle, that is this repeat region of tau, is actually binding to domain 4. There's a little bit of binding to domain 2, but it's not nearly as strong. And so what we can do now is we can, if you go over here to the right, we can actually make these mini domains. We've even made them free of the membrane and can then immunoprecipitate them and show that tau comes down with the LRP1 mini domain, with domain 4. And this is now turning out to be a very, very nice assay. We've been collaborating with some people like Steve Olson at Sanford Burnham to develop some small molecules that might be specific for that interaction between tau and domain 4, because certainly inhibiting this entire receptor is not a good strategy because it just does too many things. So here we go with onto a model system here to finish up this piece of the talk. We think that tau is beginning up here in the upper right, where you see it's coming out of cells, and we don't know too much about this exocytosis mechanism that comes out of cells. It's in the extracellular region here, where you see tau now in the blue square, and the form of tau there is pretty much unknown. Is it already misfolded? Is it already a fibril? Is it an oligomer? What is it really? What is it? But whatever it is binding to the LRP1 receptor, which is turning out to be an incredibly fascinating nexus for the whole tauopathy process. Because there's a lot of interesting other proteins in the neighborhood. ApoE, which everybody's familiar with, binds to LRP1 as well, and may have some effect on mediating uptake. And then also in the neighborhood are these HSPGs, which have, as I've already mentioned, have some effect on uptake. So here's the binding, and then it brings tau, gets endocytosed into the cell in some form, monomer, oligomer, fibril, we don't really know. Then there comes another mystery. It has to get out of the endocytic vesicle to template normal tau in the recipient cell. So there's another area that needs better understanding. So, of course, all of us in the tau field were very influenced by this impressive work that came out from Scherz and Goder, in which they now have created this whole, palette of tau folds. And remarkably, what their work has taught us is that the particular, at the atomic level, the misfolding of tau by cryo-EM, the particular type of fold corresponds to the phenotype, to the disease. There's, as you see at the very top, there's an Alzheimer fold up here. Below that is the chronic traumatic encephalopathy fold. Here is corticobasal degeneration, PSP. These different tauopathies have unique types of folds. And this I think is a very critical observation, because what it means, I'm just going to go back to this slide here, it means that depending on where the folding is taking place, somehow when it moves from here, the donor cell, to the recipient cell, there has to be some way that misfolding pattern is communicated into this recipient cell. So if it comes in as a already misfolded fibril, that might be one way in which it's templated, but somehow in that transient form of tau, the form of tau that's in transit has to be able to communicate the atomic level misfolding structure into the recipient cell. And that, I think, is a very interesting problem. What has also added to the interest level here is another discovery that came from our extended group working with my colleagues in Colombia, where Joseph Arboleda, a former graduate student of mine, now at the Mass General and trained in Colombia as well, found the Christchurch variant of ApoE in a case where of a woman who had the presenilin 1 mutation, therefore would have, in this large family we study in Colombia, and there she would have been expected to get Alzheimer's like almost everyone in her family at around age 45 or 50, but she was now over 70 and still did not have Alzheimer's, had some mild cognitive impairment, but still did not have Alzheimer's. So it was a mystery, and she also happened to carry this variant of ApoE3 called the Christchurch variant. Over on the right, you can see where that variant is at position 136. And now we have a paper under review in which this woman who recently died in her mid-70s, not of Alzheimer's, she had a melanoma. She died, she generously donated her brain. And here is the results of that autopsy work done by another colleague in our group named Diego Sepulveda. And he he's found that not only was, in this case, was that this woman, although she had massive amounts of amyloid, she didn't have very much tau spread in her brain. Somehow the tau spread was being prevented, whether that was due to ApoE is the Christchurch variant is an intriguing hypothesis for which we have no evidence at this time. But of course, we're studying all that. Here's the data down here in which you can see that here's her frontal cortex full of A beta, but very little tau. She did have tau in the hippocampus. And interestingly, she did look a little bit like some of these forms of Alzheimer's in which you have a little bit more accumulation of tau in the occipital cortex. She had tau accumulation in the occipital cortex, although did not have a lot of higher level visual impairment that we were aware of. Anyhow, remember that ApoE is part of that complex around LRP1. So maybe this resistance to the disease that this woman had, if it was related to the Christchurch variant, could have something to do with inhibiting spread and uptake. Of course, I just want to say one more word about this very important word of caution. Remember that this is one case. It's a case that has gotten attention justifiably because it's just so intriguing. But one individual with a variant is by no means proof that the Christchurch variant is causing what is definitively an incredible clinical syndrome. That is having the presenilin mutation, an E280A mutation, and not getting disease till her mid-70s. We don't see that. That mutation is very highly penetrant, and we don't see escapees. So let's go on and now talk about another facet of the problem. So tau's inside the cell. What happens next? Tau may get into the cell by spread, or tau may even arise in a misfolded form within the cell itself, especially if there's mutant tau. As you know, there's a number of tau mutations that lead to frontotemporal dementia. So we can ask the question here in the red box, What is there some cellular environment that's particularly conducive to tau misfolding? Well, so here let's talk about that for a moment. I've shown here tau, mutant tau coming off the microtubule where it lives normally, and then headed for these degradation pathways, which I'll be talking about a little later. So, so here's one study that we did, which might address this a little bit. This is a paper that's also under review. Single cell transcriptomics of 4 brain organoids carrying various MAPT or tau mutations. So we have iPS cells from individuals that carried tau mutations. One of them was 337M, another 406W, which you see there. From those iPS cells, we made brain organoids in collaboration with Sally Temple, and these organoids were then studied by single cell transcriptomics. And here are the kinds of, this was done by Stella Glausser and in my lab. And you can see here the typical way in which we sort of parse out cell types in a single cell transcriptomic analysis. That is, we lay out all the transcriptomes and create in this very high dimensional way, and then create a UMAP, which now sort of visually separates their variation. That takes what's basically a principal component analysis, and then in some nonlinear way separates out all these differential transcriptional profiles of individual cells and clusters them according to cell type based on the markers that are in each one of those clusters. And you can see that we get a nice distribution of cell types here, and I'm going to emphasize the green over here, the astrocytes, because that's where we begin to see some interesting differences. And so one thing we can do is now recluster just the astrocyte pool here into 4 clusters, 0, 2 through 3, and then label every cell, not just based on on which cluster it belongs to, but also labeling cells whether they come from the control or the mutant individual. And that turned out not to be super informative. We just didn't see that one particular subtype of astrocyte was harboring all the mutants. So we had to move into another approach to this, which is looking at the entire astrocyte pool and looking for DE, differential expression of marker genes. And here's where we hit it. It turned out that there, we see here over in the upper right, there are some genes that are expressed at a higher level in this volcano plot and some at a lower level. And if we look at what the genes are, there's a few of them, but a large cluster of them fit into a cholesterol biosynthetic pathway here. Again, Stella's work. And so you can see the genes in pink are those that are upregulated in the organoids carrying the tau mutations. And one very famous gene here, HMG, that actually is very rate limiting in the pathway toward cholesterol synthesis, is among them. And not only is it among them here at the mRNA level, but even when you do immunocytochemistry on the organoids, here you can see the control. This is V337V. It's just there's no change at that site. But if the mutation is made V337M, you can see that HMG has increased expression here in those cells. So we conclude that the cholesterol biosynthesis pathway is upregulated in these mutants. We did find that the transporter, which is also interesting, I didn't show it, that the ApoE transporter is downregulated. So there is an effect on ApoE. I don't quite understand how that's going to fit yet. And then there were other genes involved in fatty acids that are upregulated. The next time we speak, I'm hoping we now have this pending. We should have it in another week or two are lipidomics, lipid profiles on these organoids, which I think is going to really tell us a lot more. So moving on, we have, I mentioned we have this nidus of action where LRP1 is operating and ApoE and a lot of cool stuff going on that may be involved, but who exactly are the culprit cells? You know, we talked a little bit about astrocytes just now. I had mentioned microglia in the region of the spread tau, and of course neurons are very crucial always. So what's going on? And so what we've begun to do is to actually look at lipid droplets that are in cells, and many cells are full of them. As we age, they tend to accumulate, and microglia have a lot of them. So in work done by Carolina Camargo in the lab, you can see here that there is lipid droplets here. These are, this is staining for called lipid tox. It's like another variant on Bodipi, which you may be familiar with, in which the red is now staining the lipid droplets in microglial cells. And these are really interesting microglial cells that I won't be able to talk too much about because it's still early days, but these are microglial cells that have been derived from IPS from humans. So lipid droplets are really, really interesting. There are probably people here who know more about them than I do. I'm just sort of wading into this because they're so fascinating. Their membranes are a monolayer, not a bilayer. So they're really, they're a very unique kind of organelle, and clearly deserve attention in this field and probably aging in general. So what another graduate student in the lab did, named Sharon, you'll see her name on the next slide. She cultured H4 cells, put them over a density gradient, developed a whole method for isolating lipid droplets. And there she is. This is Sharon Tian, and she found that when she looks at makes tau in vitro and purifies these lipid droplets, that now by adding tau, she can show that the tau appears to be entering the lipid droplet. And so here you see double labeling. Here's the merged image. Green plus red is yellow, and that's pretty much what she's seeing here in this experiment. Furthermore, she went on to then go from looking at those tau monomers to actually looking at various conformers. By conformers, we mean not just the monomer, but also oligomers and tau fibrils. And they also seem to have interesting relationships to the lipid droplets. Here, the oligomers seem to sort of become adjacent to them, and then they cluster around the lipid droplets. Tau fibrils appear to maybe even create some sort of fusion among lipid droplets. So there's a lot to learn here. Here's another picture in which oligomers may be forming, oligomers of tau in the red around the lipid droplet in green. So this is just to bring you, this is all unpublished data that's just intriguing us immensely, and I just wanted to share it. If you take this to the EM level, you can see here again, let's look at the big picture here over on the right, where you can see the lipid droplets. And this astounds me. I mean, there are like fibrils happening all over the place, you know, even at quite some distance from the actual droplets. I really wonder how that's happening. In this case, the tau oligomers were made. In fact, all of our tau oligomers have been made by with heparin, which is not perhaps the ideal way to do it. It's what most people have been using for a number of years, but we now know that I showed you that again from work from Michel Goder, that the fold that's induced by heparin on tau is not identical to the Alzheimer fold or to any of the disease states. So the field is beginning to have some skepticism about using heparin oligomers, but they clearly template tau. They do a lot of really interesting things, and it remains that it's an interesting model, but we've got to do better. One technology that we just started in the lab, which I'm just going to show you because it's interesting technology, I don't have data yet, which is this OPTIR. This is optical photothermal infrared visualization. What infrared does is it shows you the vibration in different types of bonds. So lipids are going to be double bond here between the carbon and the oxygen, and then there's amide bonds in the proteins. So this work done by Iran Bai in the lab, who recently joined us after doing some as an engineer at BU. She has developed this technology where she actually adds this photothermal approach in which the section gets heated a little bit, which changes some of the vibration, and you can now get even better resolution. Here, this is just to show you some of her work in a cancer cell, where you can actually see lipid droplets here. Remember, there's no label here. There's no Bodipia, there's nothing. This is just looking at bond vibration to pick up lipid droplets. It's an amazing technique, which we're about to apply to some of these microglial cells. So far, all we have done here, this is her data again, is that she can use the technology to distinguish in solution tau monomer, oligomer, and fibril based on the different bond conformations. So I'm going to now sort of come to the beginning of the end, and this now is what happens next. You know, this is the continuing theme of what's happening to tau when it's inside the cell already. We're no longer talking about uptake. Well, one area which has gotten a lot of attention in the last few years, a whole field opened up by Brangwan at Princeton, has to do with phase separation. In biology in general, this is really a fascinating problem in which proteins and nucleic acids, a lot of molecules will interact with each other, not by actual bonding, but by these by forming multivalent weak interactions, often mediated by charge, but not necessarily. Sometimes by other groups on some other ring structures and pi bonding, but there is mostly these are charge-mediated clusters of proteins that all are held together the way when you think of oil and water, the droplets of oil and water, they're not, they're held together as a phase separation. The oil is phase separated from the water. And this happens in biology all over the place. It's gotten a lot of attention from people like Brangwan because their background is in physics, but it's been known in biology for a while that there are many organelles in the cell, like stress granules and P bodies and even nuclear pores, the Cajal bodies in the nucleus, that are all membranous organelles that are held together as phase separated entities. And there's been work recently, including from our group, in which we can show that tau in vitro is can be phase separated. I have a slide coming up. I'll show you more about that in a moment. But I want to talk about that here too, because I'm going to be leading up to a type of degradation condensate with a marker called BAG2, in which tau will enter, and then that leads it toward degradation in the proteasome, as you see over here. The reason this has been interesting, this was sort of in the innuendo of what I've been already saying. I'm just going to the next slide to talk about this. Here is a diagram from a person who's very well known in the whole field of phase separation, Papu, doing liquid-liquid phase separation. You have to realize that just like oil and water, right? The oil too is still a liquid. So these are two different phases of liquids. And he has done a lot of work to sort of understand the physics of this, of how this happens, and how it may be modified in the presence of chaperones. Where I was going with this is over here to this more solid arrow over here, where once you go from a densely packed structure, because remember, tau within the phase separated organelle is more densely packed. So although there's proof for this yet in the tau field. This is a strong suggestion with TP43 that once these proteins are phase separated and more densely packed, they may be prone to forming fibrils. So that is that the symmetry is broken, and they would oligomerize, form an oligomer or fibrilize under those conditions. For tau, that's still a hypothesis. Here's our work to show you the work done with a very close collaborator of mine in the chemistry department named Song-Hee Han, who is showing, this is all showing phase separation of tau in vitro. It forms these very nice little droplets. And if you're a chemist and not a biologist, you tend to call this process coacervation. So here, by way of background, just one slide before one or two slides before I come back to tau to show you what lead into this latest discovery, still not published, done by Daniel Carretero in the lab, is that we had previously published a number of years ago that this BAG2, which is a well-known co-chaperone, can mediate the degradation of tau. But now we're showing also that BAG2 is a marker for a phase-separated structure in cells. We're no longer in vitro, because that's really the goal. And you can see these gorgeous looking droplets here in cells. This is the control panel stained for BAG2. And then if we stress the cell, do hyperosmotic stress with sucrose, we increase them. So these are phase-separated units that are related to stress, but I'll show you in the next slide that they're not stress granules. Here's another proof that these are phase separated, that is, they undergo fusion. We track two of them, we can watch them fuse together. And another proof over here is to frap them, that is, we bleach them and they recover. So these are all proof that we have phase separation. Unlike stress granules, these BAG2 condensates, there's no RNA here, which stress granules obviously have, P-bodies have. BAG2 condensates, they don't have ubiquitin, which is also interesting, but they do have another protein called PA28, which mediates protein degradation through the 20S proteasome, not the 26S. So when a protein has to get a protein that may be misfolded and is no longer globular, has to be threaded into the open pore of the proteasome, it seems that proteasomes that lack the cap, they're the 20S rather than the 26S, are better recipients for that type of degradation, which is what happens actually to synuclein, by the way. And it seems to be happening to tau as well. So this form of the proteasome, of the 20S, is also located in these BAG2 condensates. And here, when we treat now cells that have tau in them with hyperosmotic stress and create the these BAG2 droplets, or condensates, as they tend to be called in the field, we can watch the BAG2 condensates collect right along the microtubule here, going right toward tau to maybe pick them up and induce their degradation under this stress condition. So here is our model system. BAG2, we're calling it a degradation granule. Remember the stress granules that we know about, the one that's named the stress granule. When a cell is stressed, the cell has to shut down protein synthesis. That's the big thing that happens. This is the work of people like Peter Walter, and it's really, so this kind of response is really crucially important. When the cell is under stress, it doesn't want to be making more protein. So RNA has to be, translation has to be inhibited, except for proteins that are chaperones and are mediating degradation. All other systems are shutting down. And the stress entities that we know about all involve RNA focused on inhibiting translation. But how about the proteins? The proteins now also have to go and get degraded. And we would propose that tau and other proteins get degraded in this kind of a phase-separated membranous organelle, the bag-tube granule. So that's what is a paper that's now under review. And finally, just to come back to where I started, we have, I talked about the spread here and talked about maybe that there might be some treatment approaches here that would target these degradation pathways. I do think number 4 here that since we're learning so much about how the form of tau that is actually getting the molecular atomic level of tau, we might be able to make disaggregation small molecules. And here I'll just mention right now what I think is really the biggest mystery right now is that, we have cryo-EM on what these inclusions look like when they're fibril. And we know a lot about tau monomer, but we just keep tossing around this term called tau oligomers. And the only thing we know about tau oligomers are that you can make them with heparin, which doesn't count. So what are the tau oligomers in the Alzheimer brain? That's, I think, what do they really look like at the very fine structural level? And I think once we know that, number 4, making disaggregation inhibitors may become more of a reality. So with that, I will end. Here's our group here. Over on the right, I mentioned a little bit of work from another faculty in the chemistry department, Sang-Hee Han, and her graduate student, Michael Viggers. So, so thank you all very much. We can engage in a conversation through email since I'm not there, or on Twitter. And again, thank you for inviting me.
Great. It was just an amazing talk. And I think we cannot hear the claps, but we can see all of the claps. This is an amazing talk. So I will open the forum for questions. I think, all of them have a lot of questions. So please just unmute your mic and go ahead or put it in the chat.
I can't believe everyone's being so quiet. That was fantastic.
It was mesmerized by the talk. That's what
I'm mesmerized by it. And what I'm really trying to do, and it'll take someone like you to do this, Ken, is integrate all sorts of things. It seems to me that I was particularly fascinated by the co-localization of lipids and tau. And it seems to me that we keep getting all sorts of lipid signals from all sorts of research that we do. And I can't string them together in any kind of conceptual model. And I think it's a big piece we're missing. So, when ApoE is a lipid transport molecule. And 3 decades later, I basically don't think we still understand why it's got anything to do with the strong relationship it has with amyloid, although not so much with tau, I would say. We've got all these signals, the statin use and the high cholesterol signals in the epi data, that suggests there's something going on with lipids there. And is there any way we can put this together in some kind of?
Yeah. So I totally agree with you. I mean, that's why even my presentation was a little bit of a hodgepodge where you saw trying to add lipid droplets to tau. And we are in a very exploratory stage, but I think the GWAS studies and, it's just coming from so many angles that there's a signal there coming that has to do with lipids. We've sort of, even clinically, when you look at doing the glucose, old-fashioned glucose studies, PET imaging on brain, there's impairments in the brain as so that related to the vasculature. There's a very important piece of this that's missing. I really agree with the tenor of your question, but I don't have a good answer.
It's interesting too, because you just tied it to disease, and especially, tau-related diseases. But right before this, we were doing a CPC with Tom Montine, and we had an extensive discussion about how we could study the role of lipids and myelin in non-disease related aging. To, the idea that maybe it was lipids are actually central to the aging process that none of us have been studying for the last few decades for whatever reasons, but what's going on with brain aging even?
Yep, I completely agree. And I see another hand up here from someone else I know. There's
Yeah, go ahead, Lindsay.
Hi, Ken. Wonderful talk, and it's great to see you. Can you hear me all right?
Yes, I hear you very
Okay.
well.
So I was just thinking about, the role of microglia. I'm in a microglia lab, so obviously I'm going to go that direction. So we've seen studies where if you eliminate microglia in a tauopathy model, so where you have mutated tau, microglia actually behave a lot differently than in a model that I'm working on, where we actually just overexpress human tau, and then we have one of those familial mutations that increase amyloid production and cause plaques to form. And so my question is, you presented and talked about how heparin is obviously not inducing misfolding that we see in Alzheimer's disease. You presented that one paper that showed all those different folding templates according to different disease models. And so I'm just curious, this phase separated tau that you're looking at, is this a mutated tau or non-mutated tau? And how do we go about, making sure we're making the correct targets for tau? Because, and maybe this is another question, is does mutated tau, is it taken up and is it degraded the same way that, non-mutated tau is?
Yeah, well, mutated tau is certainly taken up. A lot of our experiments were done with P301L. So that part of your question is easy. And there is some data to suggest that tau harboring a mutation, at least in vitro, has enhanced phase separation. However, it's still, although the hypothesis is out there that forming these condensates, densely packing tau into a phase separated state, is, some people would say, is on pathway to the fibril. I don't know that's really true. I think that still requires some proof. So, but what's happening in those condensates, that the molecular interactions in there and how they're accelerated in the presence of a mutation is, I think your question is so on target. There's such complex structure. It really, the chemists have liked them because they can just take these proteins and look at them one at a time and make, just tau alone or tau with certain amount of salt, change the temperature. We can't do that. We have thousands of proteins. These condensates that I'm talking about, whether you're talking about a BAG2 condensate or a stress granule or whatever, they're made up of hundreds of proteins, maybe more. So the molecular interactions there are really, really tricky, and how that's going to be affected by a mutation, I really just don't know. I think we have a lot to learn about the relationship between what, if you have an oligomer, then there's one kind of interaction. But if proteins are forming because of weak multivalent interactions in a condensate, that's another type of reaction. And the relationship between those two, I think, is going to be very important to understand at a basic physical chemistry level. Yes.
Hi, Ken. It's Leslie. That was just a fantastic talk. I really enjoyed it. I was really fascinated by the granules, the liquid droplets that, or the phase separation without RNA in there, and having this sort of more proteasome direction. And do you think, and having BAG1 in there, or do you BAG2, right? Do you think this could be a common feature in some of the other disorders, or do you think this is strictly a tau related process? Or, maybe too early to know.
Yeah, so phase separation has had a lot of play in this topic, as you know.
Yeah.
The TDP43 has been looked at a lot, and so have some others. There's some work on synuclein and Huntington. They've all been looked at, and it's quite clear that proteins with prion-like binding domains and RNA binding proteins, this whole family of proteins, are very relevant for phase separation and the transition to fibrillization. So basically the answer is yes, this field pertains to almost every form of neurodegeneration.
Right, but I mean this where you don't find the RNA in there, because we find granules, you know,
All
that
over
look
the
like
place.
G3BP1 granules and stuff in
Exactly.
HD and
Yeah.
other diseases, but that they
Right.
might, I hadn't thought about not having RNA in them like that.
So that's the insight here that I'm also very impressed with, that, you know, that there's the cell is putting a great effort into making these types of granules with RNA so they can inhibit translation. But I think we've overlooked exactly the protein pathways. And that's what I'm hoping that the discovery will imply, that the BAG2 is granule is one entity that may be handling that quite specifically. And it's interesting that it goes right to the place where it's needed. I mean, those BAG2 granules, they travel to the microtubule. I really like to see if they, you know, it would be interesting if they travel to Huntington's disease protein or other proteins when they're misfolded.
Yeah, me too.
Yeah.
So we'll be talking. No.
Yeah, we'll have to talk some more.
I can. Really fantastic stuff, very provocative. I've got a couple of questions. The first one is tau phosphorylation. You mentioned acetylation of tau, but you didn't really mention phosphorylation. So I'm sort of curious where you think that phosphorylation fits into things, or if it's merely mediating the acetylation effects that you mentioned. And then the second part is which cells are taking up the tau? Is it every cell type that you look at, or is it specific types?
Yeah, so so one problem with phosphorylation is it's too complicated. You know, there's like 26 sites of phosphorylation on tau that occur in all kinds of combinations and permutations. Phosphorylation did not affect the uptake of tau. We did look at that. That was not affected, but there's no doubt that phosphorylation has a very important role. We were fortunate to collaborate with Judith Steen, who just had a nice paper on looking at all the phosphoforms of tau and other post-translational modifications in tau in CSF from individuals. It's a very comprehensive paper. And worth looking at. The other question about the cells that are taking it up. So clearly, I mean, neurons must be taking it up at some level, but this idea that perhaps microglia are also picking it up and spitting it out is starting to be very intriguing to me, and I think it's probably been intriguing to you for a lot longer. So I would, I think we don't have a really solid answer for your question, but ultimately it's going to end up in a neuron.
Thanks.
Yes. I see another question there. Yes.
Hi, thanks, Vivek. That was really provocative talk. Thank you very much. I had two questions. The first one kind of follows on from what Leslie was discussing earlier, and that is when the lipid droplets and the associated tau are stuck on the microtubules, I wondered if you've tried to use something like a GFP tagged KIF1A to see whether it's affecting kinesin trafficking or even dynein and retrograde trafficking of cargoes such as mitochondria or, you know, synaptic vesicles, a variety of other things, and whether that could be involved in the pathology here in the neurons.
Yeah, we've thought about that. I would love to do that experiment. Absolutely relevant and so fascinating. We haven't done that though.
Yeah, I mean, it's interesting to think about generalized defects in microtubule-based trafficking systems as being a bystander effect of this change, this flux in the lipid droplets and the tau gumming up the works, basically gumming up the highways. Yeah.
Yes, yeah, and I know there has been papers, as I'm sure you know, from the Mandelkels about, you know, the kinesin affecting tau movement on microtubules, but now to have those droplets there, to me, is very, very intriguing, because it really means that you know, we've always been curious about the on-off dynamics of tau on microtubules, right? So when tau is off microtubules, why doesn't it just float away and diffuse in the cytoplasm? Well, if it really can be off the microtubules, but live in a condensate right next to the microtubule, then it creates another place where there can be the on-off dynamic happening in greater proximity to the microtubule. So
yeah, I agree. I mean, I think in general, free diffusion, people usually think of the cytoplasmic environment as a soup, but in reality, it's not. It's
not even close, right?
It's like a rave party where there's people shoulder to shoulder everywhere, maybe.
Yes.
The other question was to do with I'm sure people are thinking of this, but I was really struck by the tau template misfolding data you showed from the Shearis group. And I'm wondering if people are doing small molecule screens to look for small molecules that would prevent particular folding conformers, and whether that might have any effect in blocking spread and or pathology.
I don't know, what everybody's doing. I have a feeling that's going on all over the place.
Okay. Thank you.
Yes.
Great. Thanks a lot. I think we have coming on the hour. Thanks a lot, Ken, for joining us. This has been an amazing talk and some vivid conversations. Please join us next week. Another Potemkin Prize winner, Dr. David Holzman, is going to be there at 11 a.m. next Thursday, the same Zoom link. So please join us for that. Thank you very much, all of you, including Glenn, for joining us.
Thank you. Right. Bye-bye.
Thank you, Ken. It's really, really great talk and good
Good to
seeing
see you.
you.
Thank you. Bye-bye.